So our understanding of tumors biology is changing over the last, I would say, decade due to the accumulation of a number, a long series actually, of indirect evidence which suggests that tumors are heterogeneous in terms of biological composition, and they are composed of different types of cells, differing in fact for their state of differentiation. And it has been proposed that this heterogeneity is supported by a specialized subgroup of cancer cells, which is endowed with the ability to self-renew and to partially differentiate. And these cells have been called cancer stem cells. So the main supporting evidence for this hypothesis, the cancer stem cell hypothesis, is represented by transplantation experiments into recipient host mice using purified subpopulation of cancer cells. So cancer cells versus non-cancer cells. And in these assays, it has been shown that the cancer stem cells are the only one which are able to reform tumors upon transplantation. The critical point is that the tumors that arise upon transplantations have the exact same biological heterogeneity than the parental tumors. So the cancer stem cells are able to reform heterogeneity. And most notably, this experiment works the same if instead of using a population of cancer stem cells, single cancer stem cells are used. So if you transplant a single cancer stem cell derived from a given tumor, the tumor that can be obtained is biologically heterogeneous as the primary tumor. So at face value, this experiment is telling us that the cancer stem cell has two properties, that of forming xenocopies of itself, a property that we call self-renewal in the context of normal tissues, and that of differentiating, so giving rise to cells which have different fate. And that's the main reason why we call these cells cancer stem cells, because functionally they behave like normal stem cells of adult tissues. So if true, this hypothesis has important implication for cancer treatment, and in fact it postulates that the eradication of the rare cancer stem cells is essential to achieve cancer cure. This is probably a critical point if one thinks to the fact that the available anti-cancer drugs have been selected for their property to reduce tumor size in early clinical trial. And this is a phenomenon which does not necessarily imply the eradication of a rare cancer cell population. And as a matter of fact, if those drugs were to be not able to target the cancer stem cell would survive as persistent cells upon treatment, thus representing the cellular basis for the relapse. And that's indeed the major problem of cancer treatment. It is in fact relatively easy to induce disease remission. However, unfortunately, in a large fraction of tumors, tumor relapse after treatment, and it has been hypothesized that the cellular basis for this relapse is indeed the persistence of the rare cancer stem cells, which might be resistant to available drugs. As I was mentioning, however, this remains an hypothesis, and there are a number of critical issues that remains unaddressed, which doesn't allow this hypothesis to become a description of the biological composition of tumors. And in fact, we don't have definitive proofs for the existence of the cancer stem cells. This is mainly due to the fact that still the definition of cancer stem cells depends on the assay you used. Based on different assays, you can score different numbers of stem cells. But most importantly, we don't know whether cancer stem cells are necessary and sufficient to support cancer growth in vivo under a called physiological situation. And this is mainly due to the fact that we do not know cancer stem cell specific mechanisms, which prevents the possibility of planning a reverse genetic experiment to show that those cells indeed, upon ablation, to show that those cells are responsible for growth and expansion of tumors. So our approach to try to contribute to resolving this issue is relatively easy. We try to purify stem cells and cancer stem cells from the same tissues, and then we try to compare biological properties and molecular mechanisms. We concentrated on the analysis of the self-renewing mitotic division of stem cells and cancer stem cells. And the reason being, as I was mentioning before, that self-renewal is the operative definition of cancer stem cells. Is the why we call it cancer stem cell is de facto the defining biological function of stem cells. It's the same for the normal stem cells, and this allows to compare directly normal and cancer within the same experimental context, the same context which define the two cells. We are using two different model systems, mice transgenic for the ERB2 oncogene, which develop mammary tumors, and mice transgenics for pMLR alpha and amyloid, which develop leukemia. And most recently, we have reported some of the work that we have done over the last 3-4 years, and which are basically two properties of the mitotic division of cancer stem cells that we have characterized. First, the fact that they divide symmetrically, which is due to loss of p53, and second, the fact that they have an unlimited replicative potential, which is due to activation of p21. So what I would like to do today is just to briefly summarize this information that we have acquired recently, very briefly, and then I would like to mention some new work that is a sort of follow-up, as you will see, of this one of these two stories. So briefly, we've shown that first that normally mammary stem cells mainly divide asymmetrically. As you know, it is known from work in invertebrates, C. elegans and Drosophila, that stem cell can adopt two modalities of division, asymmetric division, whereby the two daughter cells, one remains stem cells, the other commits toward differentiation. I will refer to these cells with the generic name of progenitor, or symmetrically, where the stem cell gives rise to two distinct stem cells. It is immediately apparent that these two modalities of division have a profound effect on the regulation of the number of stem cells. And in fact, if stem cells divide asymmetrically, they can divide as many times as they want, but the number will not increase. They will go from 1 to 1, the opposite with the symmetric modality of division. So normal mammary stem cells, as expected based on this work in invertebrates, divide asymmetrically. They are also able to divide symmetrically, but they mainly divide symmetrically 90% of times. And this is due and imposed by functional p53. The mammary cancer stem cells, instead, have the opposite behavior. They mainly divide symmetrically, and we show that is due to the loss of p53, which accompanies this type of tumors. Then we have played a little bit with the reactivation of p53 using drugs which compete for the p53-MDM2 interaction, and shown that the reactivation of p53 in these tumors, as expected based on those data, indeed switches back the modality of division of the cancer stem cells from symmetric to asymmetric. Consequently, and as expected, it reduces the number of cancer stem cells in the tumor, both in vitro and in vivo. Most importantly, reduces tumor size. I would like to stress the fact that under these experimental conditions, however, the reactivation of p53 did not induce any modification of the survival or proliferation of the bulk tumor cells. So basically, it has only an effect on cancer stem cells, which, if you wish, is a demonstration or a first demonstration that targeting specifically the cancer stem cells might lead to tumor reduction, tumor size reduction. Okay, then in collaboration with Pierpaolo Di Fiore's group and Salvatore, which I'm sure we'll talk about in great detail, they have shown that the more undifferentiated human tumors have behaviors which are similar to what we have seen in the mouse ERB2 model. So we have proposed a model based on these findings where we postulate that the alternative usage of symmetric and asymmetric divisions together with a switch from a prevailing asymmetric to a prevailing symmetric modality of division might be responsible on one side for the tumor heterogeneity, the fact that the cancer stem cell knows how to divide asymmetrically, allows that cells to generate progeny, differentiated progeny, but also tumor expansion. I have introduced in that cartoon, in the red boxes, 2 symmetric division out of 11, I guess, asymmetric division. And you see that immediately clear that this means at every symmetric division, doubling the number of cells. So rare symmetric division means a logarithmic growth of the tumor. The second phenotype, biological property of the self-renewing divisions of cancer stem cells that we have studied is their replicative potential. Clearly, the replicative potential of cancer stem cells is unlimited. And this is best shown by the fact that it's our common experience that we can serially transplant cancer in mice as many times as we want. Since the transplantation is a stem cell assay, this is telling us that the cancer stem cell is immortal. This is not what happens with the normal stem cells. And in fact, the replicative potential of normal stem cells is limited. Replicative potential meaning how many times in one lifespan a stem cell can replicate without losing its functional potential. If you do serial transplantation with a normal bone marrow, for example, you can go ahead for 6, 7 times, no longer. And there are papers recently published, actually the Trump paper in Cell, showing that in mice, the hematopoietic stem cells can replicate only 6, 7 times. 6, 7 like the transplantation. I think it's a coincidence, though. Nice coincidence. Okay, many groups, the most contributing of them, Weizmann group, has shown that this limited lifespan of normal stem cells is due to accumulation of DNA damage by the stem cell, genetically proven. And the Skaden group years ago showed that the cell cycle inhibitor P21 contributes to maintain the replicative potential. And in fact, in the absence of P21, the stem cell, the hematopoietic stem cell, does not replicate 6, 7 times, but 2, 3 times. So somehow P21 is involved in the regulation of self-renewal in normal stem cells. So we asked the simple questions like, how is that the cancer stem cell become immortal? Which are the molecular mechanisms underlying? Is P21 implicated? And when does it happen during transformation? And we used as a model system the leukemia mice. The reason being that these mice do not develop leukemia immediately, but they do it after a long latency in a subset of mice. and the leukemia emerges as a clonal disease, which clearly indicates that those oncogenes needs other genetic abnormalities to give rise to a full-blown leukemia. As a consequence, the mice undergo a pre-leukemic phase where the oncogene is expressed in the bone marrow, yet the bone marrow is normal. It's not leukemic, it's morphologically normal, but it has an increased probability to become tumoral. So we asked, when does the leukemic stem cell, the oncogene-expressing stem cells, become immortal, and how? So the results, which are published, again, are summarized in this slide, and we show that expression of this oncogene in the normal hematopoietic stem cell induces DNA damage, which leads to activation of P21, of the cell cycle inhibitor P21. This reduces the burden of DNA damage, thus extending self-renewal, conferring an immortal phenotype. Genetically, we've shown that this response to oncogenes in normal hematopoietic stem cells is absolutely critical for leukemia development, and in fact, in the absence of P21, leukemia does not progress in the case of ML1-ITO, or it does progress in the case of pMLR alpha. However, the leukemia is not transplantable, so there is a problem with the self-renewal of stem cells. Fine, this is cute. However, it is in apparent contradiction with a lot of work that many groups have provided over the last 10 years, including mine, and which showed that oncogene expression in a variety of cell types, usually in fibroblasts, oncogenes like RAS, RAF, MYC, you name it, induces DNA damage. However, this leads to activation of P53, which on its turn of the P53 pathway leads to apoptosis or senescence. And this pathway is felt to be a powerful barrier to tumor development. Not surprisingly, is executed by P53, which is the most frequently altered tumor suppressor in human tumors. And this in vitro behavior of normal cells upon expression of oncogenes is supported by, in the sense that it probably mimics what happens in vivo, and in fact many groups have shown that that pathway is indeed activated in early phases of tumorigenesis. And in fact, it has been shown in lung, colon, prostate, bladder cancer, melanoma, and lymphomas. This is at least the first 8 papers that were published, that in the early phases of tumorigenesis, you can indeed in vivo detect oncogene expression, DNA damage using H2AX phosphorylation as a surrogate marker, activation of a DNA damage response pathway involving P53 and P21, and senescence. Early phases of tumors are full of senescent cells. And it has been shown in model system that activation is critical for tumor progression. And in fact, if you delete ATM, for example, or P53, the tumor goes fast. So it is clear that this pathway, it is a tumor suppression pathway. So why we see the opposite? We do see that expression of oncogenes, leukemia oncogene, hematopoietic stem cells, like the expression of RAS, RAF into epithelial cells or fibroblasts, induces DNA damage. However, in the first case, activation of P53, sorry, that's wrong. In the first case, activation of P21 and survival, actually immortalization. In the second, apoptosis and senescence. So why different responses to different oncogenes and in different cells? So the first possibility is the leukemia oncogenes are strange oncogenes. I mean, at the end, leukemia is a particular type of tumor, but this doesn't seem to be the case. And in fact, if we express the leukemia oncogenes into fibroblasts, we do see the same that we see with RAS, P53-dependent senescence. One can think that it's a problem of the hematopoietic cells, which are also particular type of tissue. I can go into many details, however, because of time, we are accumulating evidence that P21 plays a similar role in the mammary stem cells, thus arguing that the different behavior that we are observing is not due to tissue-specific behaviors. So we are left with the alternative hypothesis that stem cells process DNA in a different way, so that the difference, the apparent discrepancy is due to the fact that we challenge the oncogene expression into stem cells, while, as by before, we had challenged the oncogene expression in more mature cells, say in progenitors. So the question arises, how do stem cells process DNA? So it is the consequence of different checkpoint regulation after DNA damage in stem cells. So we ask the question directly by studying the response to X-ray in normal mice in stem cells versus progenitor. This work has been done by Alessandra and Angelo, two brilliant fellows in the lab. And the experimental approach is simple: X-ray treatment of mice, wild type or P53 or P21 null, to then investigate the role of these two pathways. Then we do time course either in vivo or ex vivo. We add the ex vivo just to make sure that everything we observe is cell autonomous, is not due to the microenvironment. The results are the same. I will very briefly show only the in vivo data. Then we purify stem cells and progenitors according to very well established approaches, which allow to purify two populations of stem cells, long-term, short-term, and two populations of progenitors, early and late, MPPs and CMPs. And for reason of brevity, I will briefly tell you about results in the true stem cells versus progenitors. The MPP and CMP behave the same. So the first information is that, as expected, in progenitors, we do see P53 activation, P21 activation, and apoptosis. And genetically, apoptosis and P21 are P53 dependent. P21 is indispensable for the execution of apoptosis. Fine. That's exactly what we knew and what we found in progenitors. In stem cells, we use different approaches, as you will see, basically conventional immunofluorescence. We set up a quantitative immunofluorescence. We call it image cytometry, Western blotting, and you will see a number of functional assays. So first, the hematopoietic stem cell, and I apologize, I'll go fast now with the data, do not activate P53. There is less than 10% that activate P53, and the absolute level, not shown here in their 10%, is very low. Second, they do activate P21 robustly, to an extent that is comparable with that seen in progenitors and CMPs. This suggests that P21 is activated in a P53 independent manner. This is indeed the case, as shown by analysis of P21 activation in P53 null stem cells, where clearly P21 goes up. Under the same condition, as you can see, P21 is completely, the response, the P21 upregulation is blunted in progenitors in the absence of P53. Then we looked at the biological effect, and if you want, consistent with the fact that P53 is not activated, the stem cells do not execute an apoptotic checkpoint. We asked the role of P21 in that, and strikingly, if you compare the two red boxes, no apoptosis in stem cells from wild type animals in your left ear, massive apoptosis in the absence of P21, as if P21 is blocking the execution of the apoptotic process. And this correlates with the fact that in the P21 null mice, P53 now is induced. So basically, putting together this data, biochemical and genetic evidence suggests that in stem cells, P21 is activated in a P53 independent manner, which leads to suppression of P53 and inhibition of apoptosis, which is somehow different from what we knew and we learned and we observed in progenitors. The same results were obtained with the mammary stem cells. And here I just summarize a few of them. You see no activation of p53 in the stem cells. These are purified at homogeneity mammary stem cells. Activation in progenitors. In the absence of p21, robust activation of p53. No apoptosis, p21 dependent. Then we ask the question, okay, fine, this is a repressive pathway of apoptosis, but does this pathway only serve to the function of inhibiting of apoptosis or takes care of the damage? It is, in other terms, a cellular response which cope with the accumulation of damage in stem cells. That's critical. As I mentioned, DNA damage in stem cells means functional exhaustion. So does this pathway contribute to maintain the self-renewal ability of stem cells? So we measured indirectly the role of this pathway in repairing damaged DNA and in maintaining self-renewal. Repair was measured roughly by looking at the persistence of DNA damage. You see 100% of cells, of stem cells, accumulate DNA damage 6 hours after irradiation. After 2 months, basically all cells are healed, somehow activated the DNA repair pathway. In the p21 null, instead, about 35% of cells maintain clear signs of DNA damage, thus suggesting that p21 helps in repairing DNA. I would like to stress, we can discuss it later on, that in the null, the p53 null, DNA repair proceeds as well. And in these cells, p21 is highly upregulated following damage. That's competitive bone marrow transplantation to measure self-renewal. Again, you see 0.5 versus 1. This is the effect of X-ray on self-renewal. It's known. You can repair as much as you want, but damage is measured as loss of self-renewal. 0.2 in the absence of p21. So in the absence of p21, you have more DNA damage and less self-renewal potential. The same results in mammary stem cells. Okay, so basically the story becomes a little bit more complicated. Upregulation of p21 leads to downregulation of p53, inhibition of apoptosis, and the same p21, DNA repair and maintenance of self-renewal. Which mechanism here? How does p21 favor DNA repair? I'm so sorry. So we are familiar with p21. We're studying since decades. p21 is a cell cycle inhibitor. Somehow it's felt that the ability of p21 to push cells into quiescence might help DNA repair. We don't know exactly why. Maybe cells, if they don't think to grow, they may think to repair. And so we measure the effect of X-ray on the cell cycle property of hematopoietic stem cells and role of p21. Basically, in vivo BRDU, Ki67, a few hours after the irradiation, and 2 months after, numbers. So the real effect on cell number. Results were surprising. We were surprised of the results. And in fact, not only we didn't see any cell cycle restriction effect, but we saw the opposite. So hematopoietic stem cells enter into the cell cycle, double BRDU incorporation, double, little bit even more Ki67, and double the number. So X-ray pushes hematopoietic stem cells into the cell cycle. And this is again p21 dependent. And the same, sorry if I don't go into the details, for the mammary stem cells. Okay, so we are adding cell cycle entry and expansion to the list of functional consequences of the activation of p21, though here we don't know what's the mechanism. Cell cycle entry and expansion, however, do not necessarily go together. If you remember what I briefly mentioned at the beginning, if a stem cell enters the cell cycle, but the modality of division remains asymmetric, this would not result in increased number of stem cells. While we do see recruitment into the S phase, but also doubling of the numbers, which suggests that cells switch their modality of division upon X-ray from asymmetric to symmetric. So we address the question directly, and in fact, this is in red the frequency of asymmetric divisions in the wild-type cells. Following X-ray, you see there is a switch from red to white, from asymmetric to symmetric. So X-rays induces, DNA damage induces symmetric division. It is the signal for the switches from an asymmetric to a symmetric modality of division. And this is again P21 dependent. These are P21 null hematopoietic stem cells treated with X-ray. You see, like the wild type, they divide asymmetrically. They are unable to switch. So how does P21 imposes this switch from a modality to another one? We knew already that P53 regulates the modality of division. P53 present, asymmetric, absence of P53, symmetric. We know that P21 represses P53, so we asked, is the switch from the asymmetric to symmetric division following X-ray due to downregulation of P53? And that's what we did by measuring P53 activity at single cell level prior to the first mitotic division by expressing GFP expressing P53 reporter into hematopoietic stem cells. Then we time-lapsed and quantified signal, and as you can see, this is the stem cell that's up to the first mitotic division, and you see that P53 goes slowly up and then goes down, probably due to the division itself. I mean, less protein divided by 2, and go on. In following X-ray, when the division is symmetric, we see the opposite. It goes down, further down, and go ahead. So we analyzed that in many different mitosis. I go quickly to the average of the regression lines of many individual cells which have been monitored, and this is the average of the wild-type cells, goes up, mitotic division, and this is the average following X-ray. So it looks like, indeed, prior to the mitotic division, P53 goes slowly down following X-ray, and as we've shown genetically, the absence of P53 or the reduced signaling of P53 allows for the switch toward a symmetric modality of division. And strikingly, this depends on P21. These are the P21 null stem cells upon treatment with X-ray. You see that P53 goes up, almost like in the wild type, and under these conditions, cells are unable to divide symmetrically. So this is my summary slide. I actually have more than one. And this is the summary of the phenomena that we observed. P53 downregulation following P21, responsible for the inhibition of apoptosis, the entry into the cell cycle, and the symmetric switch of division modality, DNA repair, and maintenance of self-renewal. So if we now put together these phenotypes, these biological P21-dependent biological behaviors, basically following X-ray, stem cell enter the cell cycle, divide symmetrical, repair DNA, and maintain self-renewal, which would lead to the transient expansion of a functional pool of functional stem cells. They increase in number, they repair DNA, and they maintain self-renewal. I guess this can be considered an adaptive response to tissue damage. It makes sense that if you damage tissue, clearly you cannot afford killing stem cells, because if you kill the stem cells, you don't have the tissue. You can afford killing the progenitors. You make as many progenitors as you want. So if they are damaged, get rid of them. But you have to maintain a pool of stem cells. And this might be the mechanism through which stem cells cope with DNA damage, which means in this context tissue damage. Stem cell has to replenish the damaged tissue. I think that, however, this mechanism might have pathological consequences, which probably are linked to time, so not seen by evolution. And in fact, continuous rounds of DNA damage and repair over a long lifespan would lead, obviously, to a mutator phenotype. DNA repair is the mechanism through which mutations are introduced in the DNA following damage. And one would expect that over a long time, these cycles of damage and repair might lead to accumulation of mutation, which might be responsible for the increased cancer risk seen with age or with the tissue aging itself. It is known that accumulation of mutation is a feature of the aging tissues. I would like to close with the implication of this study with respect to the question that we asked. So what happens now in cancer? Cancer, as I was mentioning before, oncogenes induce DNA damage in stem cells. So what would be the consequence of the activation of P21 following oncogene expression? The number of stem cells will continuously increase. Oncogene is not like X-ray. X-ray is 1 minute, oncogene is forever, so there is persistent DNA damage. One would expect an increasing number of stem cells, would expect continuous waves of continuous wave of damage and repair, and immortalization, extended self-renewal. In other terms, and this is really what I guess might represent a description of genomic instability, because you have continuous, a chronic mutator phenotype due to this wave of damage and repair, leading at the end to an expanding pool of immortal and genomically stable stem cells, which might represent indeed the early step of transformation. Oncogene expression, you want to have a perianal cell which survive enough to lead to manifestation of tumors, which is genomically stable. How this correlate with the data we have seen in vitro, senescence, in the early phases of tumorigenesis? I guess that these data are consistent, because if an oncogene is expressed in a normal hematopoietic stem cell, that would activate the P21 checkpoint, leading to the expansion of this number. However, as soon as the stem cell will differentiate, immediately P53 will be activated, thus leading to accumulation of senescent progenitor. So the early lesion might be expected, based on this model, indeed composed of senescent cell, but with the presence of surviving and immortal cancer stem cells. Clearly, this is due to two different checkpoints. Here, the oncogene activates a P21 checkpoint. Here, the oncogene activates a P53-dependent checkpoint. Clearly, this situation creates a pressure for loss of P53. And when this happens, one should expect the progenitors evade the senescence program, thus proliferating, and this might be one component of the switch from a benign tumor containing senescent cells versus a growing tumor. Clearly, this is an oversimplification. Many other things have to happen, but we have time, no? I mean, we are young. Thank you very much for your attention.